Today's lecture is on genome-wide association studies. These type of studies are used to correlate the data that is produced by SNP arrays or by whole genome sequencing with different diseases or different traits. So basically, you're correlating specific gene variants with phenotypes that are present in a specific population. This entire lecture is tailored towards helping you decipher the paper that you need to present in your poster presentation. It shows you how GWAS is designed, how it is executed, how data is analyzed, what are the biases and limitations that could possibly arise in such a study. So at the end of the day, you will be able to read a paper and be able to put together a full presentation just based on the information that you are given today. As for the learning objectives of today's lecture, you need to know the different types of genetic analysis. Once we have the big picture, we focus on genome-wide association studies, what they are, what the general study design is for all GWASs, what the different limitations are. Basically, we're talking about population stratifications and the different methods employed to circumvent it. How the data that is obtained from a GWAS is actually analyzed, and how additional SNPs are inferred based on haplotypes. Also, you need to understand a little bit about the limitations of GWAS. So let's see now how genotyping information and data from whole genome sequencing or from whole exon sequencing can be used to determine variants that underlie or contribute to either human diseases or to certain traits. The goal here is to basically establish a catalog of all the genes with all their functions, their structure, their location on the chromosome, and basically to add to the growing list of millions of variants between the different, among different individuals in different populations. Some of these variants are functional, so they actually have a big impact on a phenotype, but most of them are neutral. Some of them are very common. You can see them present in almost all individuals, and some of them are very rare, restricted to either families or to small ethnic groups. There are basically three major approaches to genetic analysis. First is linkage analysis, which is not that often used nowadays. It basically uses family pedigrees to follow the inheritance of disease in certain family members. And it basically looks at the role of a specific variant or specific variants that could actually correlate with the disease in that family. It is an approach that is called a candidate gene approach, where basically, based on the phenotype that those individuals have, a person can infer one or multiple genes that could be targets, that could be distinctly set as targets and responsible for that phenotype, so they are set as targets for analysis. This type of linkage analysis is mostly used for traits or disorders with Mendelian inheritance. The second type of genetic analysis is association analysis, which does not rely on pedigrees, and it rather looks at entire populations with a specific phenotype for increased or decreased allele frequencies when compared with a control population that does not have that particular phenotype, whether that is a trait or a specific disease. So this type of association analysis are usually used for complex multifactorial diseases or traits. The third approach is a little bit more in-depth and more convoluted, and it relies on the data produced by genome-wide whole genome sequencing or whole exome sequencing. By sequencing the genomes or exomes of affected individuals and their immediate family members, it is extremely important to very, very rare Mendelian disorders where linkage analysis is actually not possible because not enough family members or not enough individuals in a population display that particular trait or that particular disease. So these are the three major types of genetic analysis. What we will be focusing on in today's lecture is on association analysis. The methods used to find variants are determined by the frequency of the alleles and the size of the phenotypical outcome, as you can see in this graph here. Shown on the x-axis, you can see the frequencies of the risk alleles, where you have common alleles with a greater than 5% frequency, down to very rare alleles, which are confined to certain individuals or certain families, and even spontaneous mutations. On the x-axis, you have the size of the effect of those alleles on producing a certain disease or a certain trait. So, for example, alleles that have a high effect size are more likely to cause a disease versus the ones that have only a moderate or low effect, which are generally contributing to the risk of developing the certain disease. So they're just increasing the risk of developing a disease. Most genome-wide association studies study common variants, as you can see here, that have very low effect or low effect. Basically, they just slightly increase the risk of developing a certain disease. Common variants with a large effect can also be identified by GWAS. However, there are very few variants that have a strong impact. One of those is exemplified here by ApoE1, which is responsible for increasing the risk for Alzheimer's disease. When it comes to very rare alleles, or the ones that are specific to just population, to just a family, the best information is given by whole genome sequencing or whole exome sequencing, which are generally necessary for detecting very low frequency variants that can then be analyzed and correlated with very rare phenotypes. So what are genome-wide association studies? Basically, the principle of a genome-wide association study is to look at a large portion of variation in the human genome and search for alleles that are associated with risk of disease or a variation in a certain trait. The beauty of these association studies is the fact that they are completely unbiased in respect to the function of the variant. Therefore, you don't need prior knowledge on the gene function, and it does not rely on the assumption that certain genes would correlate with certain phenotypes, as is the case with the candidate gene approaches. So, genome-wide association study is basically an approach that involves scanning, a very superficial and rapid scanning of markers across complete sets of DNA or genomes of many people in order to find small variations that are associated with a particular disease or a particular trait. Unlike the candidate gene approach, which we saw in the linkage analysis, which is actually more powerful in terms of the information that you get and the strength of the statistics, that one is generally confirmatory of previous knowledge, versus GWAS, who takes a global exploratory approach without the need of any prior hypothesis on the function of a specific gene or variant. So once a new genetic association is identified, the next step is for researchers to use that information to further characterize the functionality of the variant, basically to determine the underlying mechanism of action for its contribution to a certain disease. Also, they can characterize the relevance or the impact of that variant towards the general population. Basically, what we have learned so far is that few of the strongest associations reside in coding regions, and the majority of the associations are actually present in regulatory elements such as promoters, enhancers, silencers. Also, for long-term goal, they can look into the commonality of that allele in specific populations, or the relevance of that allele for risk of disease. And using that information, they can build predictive models that associate a specific phenotype with a specific genotype. So all study designs, all GWASs employ the same study design. In general, for study designs for complex traits and multifactorial disorders are either cohort studies or case control studies. The majority of studies for GWAS are usually case control studies, where you actually have a case group with people that display a certain phenotype or have a certain disease, with a control group that does not have that disease. The next step is to select a platform for genotyping, a SNP array. After that, there's a need for reduction in the, or for assessing for the quality of both the sample and the SNPs. All those that are not of very good quality, they need to be discarded. And once that is done, then the GWAS data is actually analyzed, and what you usually get is this Manhattan plot. After a GWAS is conducted, the next step is to be validated by different studies, which can be then combined into a meta-analysis. The biggest challenge in a case control study is usually to find cases and controls that are very similar in absolutely every way. Age, sex, demographics, health status, ethnicity, everything except for the disease or the trait in question. So generally, the way you select cases, if you want to enrich the effect of the genetics, then you select individuals that have a more severe phenotype, or the ones that have the disease at a younger age than they are supposed to, or the ones that have multiple family members that are affected. Also, you can go by selecting the controls so that they have lower risk for disease than the general population. An important aspect is to match individuals based on ancestry, since allele frequencies between different populations are very different. So this is what we call population stratification, which can be a big confounding factor when establishing a GWAS. So let's take a closer look at population stratification. What is population stratification? Is when you have in a population, you have multiple subpopulations. And there is a difference in the representation of those subpopulations between the control and the cases. So, for example, if you have an overrepresentation of those subpopulations, as you can see in this case here, in the control versus the case, if you have an overrepresentation of those subpopulations in the case versus control or vice versa, it can lead to false positive associations that are based solely on ancestry and not necessarily on the association with the disease. For example, many traits or many diseases are very strongly associated with a certain population. Let's take the example of Crohn's disease. For example, in Israel, Crohn's disease is way more prevalent in Ashkenazi Jews. Now, if you want to conduct GWAS in Israel, chances are that cases that you will select of individuals with Crohn's disease will likely be from that subpopulation of Ashkenazi Jews. However, the controls will probably likely not be. And will have a different ancestry. In that case, that means that the percentage of Ashkenazi people in the cases will be way higher than those in the control because of the high prevalence of disease in that particular population. So, genome-wide association studies will end up showing that every SNP that is specific to that ancestral group is statistically associated with the disease only because of the structure and the stratification of our sample, and not necessarily because it has a real correlation with association with the disease, where it can change the risk for that particular disease. So population stratification is a big problem, and it needs to be addressed in different ways. The most obvious one would be, well, just avoid it, and that means carefully match the controls and the subpopulation and the cases to avoid any kind of mismatched subpopulations. Or you can restrict your entire study to just one very narrow ethnic group. The other way is, well, if you have already set up your cases and controls, you can account for this population stratification based on the fact that ancestry usually can be inferred from the genotype data based on certain common markers that are specific to certain subpopulations. And after that, there can be, you can adjust for those factors in the statistical analysis. Basically, you can either select specific statistical tests that eliminate those differences, or you can remove the outliers, or you can control for variation. There are different ways. We're not going to go into the details of statistical analysis, but there are different ways for a good and solid study design to account for the population stratification. So once we have our cases and controls, they need to be genotyped. Genotyping is the most efficient and cost-effective way of generating large amounts of genetic data, since it uses a large set of variants that are present on the genotyping array. An array can hold anywhere between hundreds of thousands to millions of pre-selected variants. I think the maximum is up to 1.5 million SNPs per array. And the two major platforms that are used currently are produced by Affymetrix and Illumina with very differently designed arrays. So for example, you can have chips that have absolutely random SNPs, some that have mostly low frequency SNPs, some that have only SNPs that are present in the coding regions, some that have SNPs in certain haplotypes, and they're called tag SNPs. And based on those specific tags, you can infer the rest of the haplotype. And also, you can also have customized arrays where you can actually add the SNPs of your own interest. So there are many choices, and depending on the frequency of the variants in a specific population, some might be better suited than others to answer a specific question. So before you actually embark on doing a genome-wide association study, you need to be very careful in selecting the right array for that will help you identify a particular association. So once the genotype data is generated, there are a few quality control measures that need to be taken into consideration before actually analyzing the data. There are two different strategies for quality control. One in terms of eliminating poor quality samples, and the second one eliminating poor quality SNPs. Let's take a look at the first strategy. What are poor quality samples? There are those that have a success rate of less than 95%. For example, if less than 95% of variants were successfully genotyped for that particular sample, that means that the sample has a low quality, and even the genotypes that, even the variants that have been obtained from that sample are likely not real. Also, you detect an excess of heterozygotes when comparing to what Hardy-Weinberg would predict, that providing that you are in areas outside of advantageous heterozygote selection. Also, you can determine that certain samples are duplicated. For example, when one person participates in a study twice and donates his DNA twice. Or you can determine that there are immediate relatives that are participating in the study. There is a pairwise matching of all SNPs, and all the immediate relatives are excluded from the study. And also, happens quite frequently that samples are mixed up, and you can notice that because the genotype shows a different sex than what the records are for that particular sample. Again, that needs to be discarded. Now, the second strategy is to identify and discard low-quality SNPs. Those are the ones that have a success rate of less than 95%. So, for example, if you have a SNP that has less than 95% genotypic success across all individuals, that SNP needs to be discarded altogether. Also, purposely duplicated samples can give different genotyping results, and that means that you have no consistency between the replicates, and those samples, those SNPs again need to be discarded. Or if the genotype frequency is not matching the allele frequency, that's another big question mark, and it indicates that those SNPs are not of good quality. So once we actually ensured that we have the best quality samples, the best quality SNPs, we can move on to associate SNPs with particular disorders. So basically, we can start addressing the question of the study that we designed. And we do that by producing odds ratios, which determines basically the association between the disease and the genotype in case control studies. Basically, what odds ratios look at are the number of cases and the number of controls that carry each one of the two alleles at a certain locus, and estimates the amount of risk for that particular variant. Basically, we need to calculate the odds of the people that have that genetic variant to develop the disease, and the odds of the people that do not have that genetic variant to develop that particular disease. The odds ratio for the disease is the ratio of those two odds, basically. So if you have a value that is larger than 1, that means that there is an association between the variant and the disease. If it is 1, there is absolutely no association. Basically, the odds ratio represents the risk of disease for a certain variant. So the higher the number it is, the higher the risk for that particular disease to be, that particular variant to be associated with a disease. So let's take a look at an example here. We have two variants at the prothrombin locus. We have G, which is the major allele, and A, which is the minor allele. So we are looking at cases with cerebral vein thrombosis and controls that are free of disease, and we are trying to determine the odds ratio of a particular allele to be associated with that disease. So what we can say based on the calculations, we can conclude that individuals that have the A allele have a 7 times greater odds of developing the disease than those who have a G, the prothrombin locus. This is how you generally find the second graph is how you generally find odds ratios displayed, especially when it comes to combining results from multiple studies. So you have the magnitude of association, the distance away from the vertical line, which is set at 1, and the confidence interval, which is basically the horizontal whiskers. Generally, there is a summary of odds ratio that takes into consideration the odds ratio for all the studies and determines an overall odds ratio for an association for a risk of disease. So before moving forward to analyze the general data that is produced from a genome-wide association studies, we need to show that there are no confounding factors at work here, and we need to be able to eliminate them if they actually are. And for that, we need to use a QQ plot or a quantile plot, which are plots that you will see in the majority of the published genome-wide association studies. And what these plots show is on the x-axis, the expected distribution of association across the million SNPs compared with the observed association, which is the one on the y-axis. Basically, a clean quantile plot that is very useful for a genome-wide association study should show a solid line that matches the diagonal until it sharply curves at the end, and those are representing the trace outliers are representing generally a very small number of true associations among the thousands of unassociated SNPs. So you can see in this graph there are different scenarios. For example, in A is a valid model, but it finds no association, no associated variance. Basically, no more appear statistically significant than you would expect. In B and C, you find big problems. Those are problematic models that show a big statistical significance for a very large number of variants, which is probably the result of population stratification. And D is pretty much what you hope to see in genome-wide association studies, that the majority of variants have no statistical significance between the cases and in association between the cases and the controls than you would randomly expect. But there is a handful of variants that you can see tapering towards the end of the graph that have an increased significance, indicating that these variants are actually associated with disease. These plots are merely helping with confirming that we have a well-designed study and that the associations we obtained are not false positives. It does not necessarily give an information of the associations between the variants and the disease. For that, we need to move on with analyzing the data that we produced in this study. So that genome-wide association study data analysis generally produces this very typical Manhattan plot, which basically resembles the skyline of Manhattan. And each data point represents the strength of the correlation between a SNP that is localized on a certain spot on the chromosome with the disease. So you can see on the x-axis, you basically have all autosomes with their tested variants, and on the y-axis, you have the p-value of their correlation with the disease. In this case, you have only one that is statistically significant. And why do we set up the statistically significant, the p-values that high? We will see next. So why do we need such stringent correlations for significance in the case of the association between a variant and disease in genome-wide association studies? Basically, the answer comes from the fact that each SNP is an individual test. So you will end up with multiple tests, a million tests if you're looking at a million SNPs, which need to be analyzed parallelly. So what would happen if, for example, you would use a p-value of 0.05, which is what is considered statistically significant in a normal research study. So in that case, with a p-value of 0.05 and a million SNPs, you would get 50,000 false discoveries just by chance in that million SNPs. So that means that there needs to be a more stringent significance in order to be able to publish anything produced by a GWAS. And in this case, it was set at a p-value of 5 × 10 to the minus 8. So this is the standard now for which at which you can actually consider that a variant is associated with the disease. So once we have all this information, we determined, like you have seen in the previous slide, you determined one SNP that is associated with the disease. Now, in order to actually make sure that those are real positive results, we need to conduct, the study needs to be conducted in a similar but completely independent group, and the outcomes need to be replicated there. Even if you don't replicate the magnitude of association, at least you need to be able to replicate the type of genetic variant that is associated with disease. But before we move on to replication studies, we need to take a look at another way to gain even more information or even more data from a genome-wide association study. And for that, we need to understand the concept of linkage disequilibrium, which basically underlies the imputation of ungenotyped SNPs. So what is linkage disequilibrium? Linkage disequilibrium is a non-random association of loci due to very close location on the chromosome, where the rate of the recombination is actually quite low. So when a SNP is correlated, when certain SNPs, 2-3 SNPs, are correlated, they are said to be in linkage disequilibrium. Basically, linkage disequilibrium is the measure of the tendency of some alleles to be transmitted together in generations as haplotypes. Those SNPs are generally very closely located on the chromosome, and that's one of the reasons why they are transmitted together. The process is extremely valuable for genome-wide association studies because it allows to infer different haplotypes based on only one or a few SNPs present in each haplotype. So because of the concept of linkage disequilibrium and this property of SNPs to be linked together, we can basically have the freedom of measuring only a subset of SNPs instead of measuring the millions and millions of SNPs in the entire genome for each genome-wide association studies. So basically, due to linkage disequilibrium, we can only measure a few and use the information, extrapolate the information that was generated from a catalog of genotypes, such as the HapMap, to add more data points and increase the statistical power of a genome-wide association study. The process of imputation of additional SNPs relies on dense genotype data that is available from this catalog, the HapMap, which we will talk about in the next slide, and the property of linkage disequilibrium relationship between different SNPs. And thus, through imputation, we can predict genotypes for a large number of SNPs that were actually not measured experimentally. So what is HapMap? I think we actually talked about it before. HapMap is a catalog of common variants across genomes. There are four geographically distinct groups that are represented: Europeans, West African populations, Chinese population, and Japanese. For all these populations, millions of SNPs were collected and characterized, along with the linkage disequilibrium patterns across the genome, and they were made publicly available. So now, based on the known variants in a specific population, we can estimate the coverage of a particular SNP, and we can input additional SNPs based on the tag SNPs that were used on that particular array. This triangular-shaped graph is generally represented in a genome-wide association study when it comes to discussion on imputed SNPs. Basically, let's take the example of a SNP that is present in a gene that has different phenotypes. For example, for for the AA gene, homozygotes, and a different phenotype for AG or GG. Let's take an example of SNP 8 in this case. You have either A or G. Since SNP A is in strong linkage disequilibrium with other SNPs on the same chromosomal block, for example with SNP 6, 7, 9, and 10, and any of these can actually serve as a proxy for SNP 8. So, for example, if an individual is TT at SNP 7, he will have the same phenotype as those with AA in SNP 8. If the SNPs are correlated, or they are in linkage disequilibrium, the trait or the phenotype is also correlated. That's why genome-wide association studies need to survey only one or very few SNPs in each haplotype block, and the rest of the data points can then be inferred from there on. So although you produce a very small number of, you look at a very small number of SNPs, you have the data from a large number of SNPs based on this linkage disequilibrium properties. So we've seen before that once we actually produced data from a genome-wide association study, so once we have an association of a certain variant with a certain pathology, we cannot stop there. We need to move on, and we need to be able to confirm that those associations were actually real. Basically, we need validation in order to weed out the false positives from the true positives. And for that, usually, it happens in the multi-stage replication approach, which usually protects against false positives and minimizes the chance of false negatives. Generally, a study starts with a smaller sample size that uses an array of a SNP array that covers a full set of SNPs, so as many as possible. And usually the p-values that are used for statistical association are more relaxed than what you would need for a general genome-wide association study. And what those things are used, the data that are produced by this, in this stage, are basically used to pinpoint towards possible candidate SNPs in the entire array. However, this is not it. We need a second or even a third validation study. And those studies have increasingly larger population sizes. And their p-values are way smaller. So they have a more stringent statistics. And also, they are not looking at full sets of SNPs. They're looking at very localized, limited areas that are only surrounding the SNPs that have been found, the candidate SNPs that have been found in the previous step. So in the case of these studies, you have large statistical power, And their Manhattan plots are also a little bit different because they represent a very limited area of a specific chromosome, particularly in the region where the candidate SNPs from the previous approach is residing. Basically, what happens next is a joint analysis, which basically combines the data from the multiple stages to increase the statistical power of the study. And for that, you need to use the methods of meta-analysis. Once a SNP is validated in this way, it is generally regarded as a conclusive locus of association with a particular disease. There are a lot of critics of genome-wide association studies, mainly due to the large number of studies that have produced basically a minimal number of significant association when compared to what was expected by now. And even those significant associations, they generally have a very modest change in the risk for disease. However, there is value in the information that is produced by a well-designed and well-executed genome-wide association study. First of all, we need to think that odds ratios are a measure of the impact of a particular allele on a complex pathogenetic pathway. So, a lot of different studies, genome-wide association studies, looking for different autoimmune diseases, have discovered that the same genetic variant is associated with different types of autoimmune disease, meaning that there is a common pathway that all the underlying all those different autoimmune diseases. Also, even if the size of the effect is quite small for one particular variant, so even if you have an odds ratio of 1.1, most of the diseases are highly polygenic, so they involve thousands and thousands of variants. And if those variants have a small effect, that effect is additive. So you will get a stronger effect once you actually have covered the entire, all the variants that are associated with that disease. So because of that, genome-wide association studies still remain an important tool to assess the genetic contribution to very complex disease. And it's important to even though there's a lot of information that was produced already, there are still millions and millions of SNPs that have not been associated yet with any kind of pathology. So basically in this, on this slide, you see the major questions that you need to ask when you're reading a paper on a genome-wide association study. And I think this will help you a lot in putting together your presentation and also in analyzing the data and in determining the limitations, the biases of the study. So in basically in criticizing the study design and the data analysis.